Greetings, attendees, and welcome to the Good Food Conference Tech Track on Production Equipment Innovation, Extrusion, and Beyond. My name is Ryan Dowdy, and I'm the Good Food Institute's Science and Technology Analysis Manager. I'll be moderating the session, and we will be focusing on better ways of doing extrusion to propel plant-based meat production and on alternatives to extrusion for texturizing and structuring. Next slide, please. Today's session will consist of 3 parts. First, we'll have 3 flash talks from Reine-Marie Guillermic from the Coxwell Group at University of Manitoba, from Valerie Pietsch of ThermoFisher, and from Ciara McDonnell of CSIRO. Second, we'll have 2 longer talks from Shannon Hood-Niefer of the Saskatchewan Food Industry Development Center and Theresa Rothenbücher of Revo Foods. Lastly, we will have a panel answer key questions on future directions for plant-based extrusion and extrusion alternatives for texturizing and structuring. We'll be joined by Martinez of Aarhus University, Girish Ganjyal of Washington State University, and Birgit Dekkers of Revo Foods. Next slide, please. We ask that all attendees follow our participation guidelines. First, be present and engaged and practice active listening. Second, speak courteously to others and respect the ideas of others. Third, participate in the session chat. Not all comments will be responded to, but all will be recorded and acknowledged. Please note that any comment, verbal or written, that is meant to attack or intimidate another person or is obscene will not be tolerated. If you experience technical difficulties, please exit the session, navigate to the Expo tab, and visit our help desk. First up, we have our first flash talk. This is from Reine-Marie Guillermic from the Coxwell Group at University of Manitoba. She'll be presenting on monitoring plant-based meat analog texture with ultrasound. Welcome, Reine-Marie.
Good morning. Thank you very much, Ryan, for the introduction. And thank you very much for having me today to speak about monitoring plant-based meat analog texture with ultrasound. I'm from the University of Manitoba, working with Dr. Coxwell, and this project has been financed by the Good Food Institute. And so you can go forward. So our goal here is really not to talk about extrusion per se, but to speak about an online quality control tool that we develop in our lab. So we produce meat analogs with an extruder, and we are interested in knowing how we can, in fact, monitor production of meat analogs using an ultrasonic device. And the question we are asking ourselves is, what are the ideal processing conditions to create the perfect meat analog and to control quality online, so the consistency of the product, for example. You can go forward. So we are using ultrasound to do that. And so why are we using ultrasound? In fact, it's a very good tool for quality control in general because it's non-destructive and it has already been used to assess the mechanical properties of materials. It's been used, for example, in NDT to detect, for example, cracks in materials and stuff like that. But here we are using it to probe the mechanical properties of our food system. Because ultrasonic propagation depends on material properties such as the density, mechanical properties, and also the presence of bubbles or scatterers or things like that present in the system. So when you send a sound wave, like you can see on the right, the sound wave will be reduced when it goes in a material because you have attenuation of the sound wave. And the velocity will be changed also because the sound is propagating in another material. So if there is a change during production that affects the mechanical properties of food, there is a good chance that ultrasound will get it, that the ultrasound propagation will be affected. So you can go forward. So like we are using it because, so it's not destructive at all. We can use, in fact, ultrasonic sensors like transducers that are put in air. So really like the wave propagates in air, then in the extrudate, then in air again before getting acquired by another transducer. So you don't touch it. It's absolutely not contact. It's also fast and not expensive, and it can see things in opaque materials that you cannot see with your raw eyes. So you measure the sound velocity and the attenuation, which are two properties of the wave. And our goal here is to do research and understand how we can relate that to texture. So basically we do also texture measurement, and we measure the texture of our material afterward in the lab. For example, the cutting peak force when we cut the material. So you can go forward. So, like this is the picture of our extruder and of our system on the left. So you can see on the left two cylinders that are in fact the transducers, and the material going in between on an inclined plane. And so it's produced with the extruder that you see on the right. And so we can change the different parameters to analyze how the parameters are changing the ultrasonic signal. You can go forward. We did that with different things, but what I will show today is an example with soy protein blend and gluten. It has 73% protein content. And we measure the peak force in the lab, and we measure the ultrasonic signal. And what I'm showing you is that the peak force changes with moisture. So basically, when you increase the moisture, the force needed to cut the material decrease. It's a bit easier to cut. The velocity change also the same way. So it's also decreasing with moisture. So basically, when you increase the moisture content, you will have a lower velocity. What is in I put here two different temperature of the barrel. Look, when we extrude at different temperatures. So one is 130, 140 degrees. And so it works for both. So you can go forward. And so the results we obtain that is interesting is that you have a correlation between velocity and force. They vary the same way, so you can really correlate the velocity that you get in your material, so the ultrasonic signal, and the force. So it's very good because it's really a potential tool to see if the material has basically the softness that you desire, for example. You can go forward. And another possible application of that is to spot inconsistencies in your material. Here I'm showing just a short example, like in blue, you have a signal that we get with like a regular extrudate in the lab with the formulation I shown before. And you can see that it has a lot of peaks and the signal is pretty large. But sometimes what you get is something like the red curve. And so it's a very much lower velocity and a much lower attenuation. So what it means is that the material is different. And those two signals were obtained in a piece of extrudate where the steady state was not reached during production, and we were not able to see that, in fact, by eye, but the ultrasound was able to pick up that there were a difference. And in fact, what happened is that you had little bubbles present in the meat analog at that time. So, yeah, you can forward. So I will conclude now. So we saw that there is a correlation between the ultrasound velocity and the texture of meat analog, and that it's possible to use it to spot production problem and material inconsistency. And our goal later is to monitor also how fibers are formed with ultrasound, and also to connect with nutritional properties such as amino acid composition and protein digestibility and this sort of thing. So you can move one forward. So but thank you very much first for your attention. And if you want more information about how we do things, you can visit our booth that will be open after the session. And you can also send us an email to Phyllis Coxell or to me, depending on what you prefer. You can also send us to both of us. So thank you very much, and have a nice day and enjoy the session.
Phenomenal. Thanks, Reine-Marie. And this reminds me very much of how geologists use ultrasound to see what's underneath the ground. Very much in the same way, ultrasound can be used here to detect and monitor production systems so that there's real-time monitoring of the texture of plant-based meats. Next up, speaker number 2. Our second flash talk is from Valerie Pietsch of ThermoFisher, presenting on the extrusion of plant-based meat. Please welcome Valerie.
Welcome to my talk on extrusion of plant-based meat, in which I would like to give you a brief overview on current equipment innovations that ThermoFisher offers for product development and material testing at small scale. But first, let me introduce myself. My name is Valerie Pietsch. I hold a Ph.D. in food process engineering with focus on plant-based meat extrusion. And in my function as application specialist for food extrusion at ThermoFisher Scientific, I support our customers in finding small-scale extrusion solutions for product development and material testing. Now, before I start talking about our equipment innovations, I'd like to start with some introductory slide. I'm sure that most of you have already noticed that the demand for plant-based meat alternatives is increasing immensely and that more and more products are actually filling the grocery store shelves. So to support this trend, it is most important that consumers that buy a plant-based meat product for the first time will like it, enjoy the taste of it, and decide to buy it again. However, here, various consumer surveys reveal that available products often do not meet consumers' expectations yet, and this is with regard to several factors that are displayed on the slide, which include the variety in texture and protein sources, as well as the comparability of retail price to meat. So to support public acceptance and cope with these expectations, the properties of plant-based meat products need to be tailored to meet consumer preferences, new ingredients need to be developed, and production processes need to be optimized. So to produce plant-based meat products, extrusion technology is generally considered as key technology because it offers a great potential to tackle the before-mentioned challenges that consumers are still facing. And this is mainly because extrusion comprises a high range of adjustable process parameters that allow to achieve different textures. You can see, for example, on the 2 pictures displayed on the slide, 2 different extruded products that were made with actually the same formulation, and actually just 1 parameter needed to be changed to gain such different results. Furthermore, another advantage about using extrusion technology is the fact that it is a continuous process that can be run at high capacities, which could in the long term also allow to tackle the challenge in reducing retail prices. However, with high degree of flexibility also comes a certain degree of complexity. And this is why we're still facing some bottlenecks when using extrusion technology to optimize the product properties. This is particularly the case for the protein texture formation, which involves a complex and not yet fully understood structuring mechanism. And to make it even more complex, these mechanisms are moreover individual for every protein source. So to gain more knowledge and confidence with your product and formulation, intensive product development studies are usually required. For many companies, however, whether it's a startup, an ingredient manufacturer, or an established B2C company, such studies can be very challenging because either they don't have an extruder at hand or they only have access to a large production scale machine where these trials can be very labor intensive, time consuming, and costly. Furthermore, looking back towards material testing for protein sourcing, these tests are very often limited to low material quantities, especially at an early development stage during protein extraction, for example. And this cannot simply be done at large scales. So to deal with these last 2 points and to make the life of our customers easier for product development and material testing, we have developed small-scale twin-screw extrusion solutions, which are displayed on this slide. So unlike production scale extruders, our extruders are easily operated by just 1 person. They require very small sample volumes of less than 2.5 kilo per hour and are scalable to larger sizes. And these features enable our customers to save time and money on application testing and accelerate their process and material development, as well as shortening the time to market for their products. So most of our customers that decide to buy our machines do this because they like the concept of being able to develop their products and test their materials at small scale, to gain process and material experience, and of course also to train their operators for large scale machines. Furthermore, I'd also like to highlight that we also offer a modular cold slit die design, which is for the extrusion of plant-based meat just as important as the extruder is itself. And this is because the specific flow pattern that prevails in a die is supposed to impart a characteristic anisotropic fibrous meat-like structure to the final product. So the most important product characteristic, actually. And since this mechanism of share-induced structuring is individual for every product formulation, of course, we put a strong emphasis in designing a modular die that provides a customer a flexible toolbox. So this way, our customers stay flexible and they can adapt the cold slit die design features according to the requirements of a specific product formulation. And last but not least, it's of course not all about extrusion. When developing a plant-based meat product, extrusion may be a key technology, but after processing, you always need suitable analytical equipment to characterize and compare the properties of your samples. And here, ThermoFisher Scientific does not only support the needs of our customers by offering small-scale extrusion equipment, we also provide solutions for the complete workflow of plant-based meat product development. So starting with extrusion systems for material testing and sampling of prototypes, our product portfolio also covers analytical equipment that can be used to visualize the characteristic product texture, for example, with our Phenom desktop scanning electron microscope, and to quantify the changes in protein structure via various spectroscopic equipment, such as UV-Vis, FTIR, and Raman spectroscopy. And furthermore, we also offer rheometers, such as the HaCa-Mars IQ rheometer, which you can use to quantify the difference in the texture of your products. Now, with these last insights into our newest equipment innovations, I've reached the end of my talk. In case you're interested in a more detailed overview on our portfolio for plant-based meat development, please visit my on-demand webinar, which is accessible on our website. And for those of you who are located in the US, I'd like to kindly invite you to our symposium on extrusion of plant-based ingredients, which we have organized in cooperation with Iowa State University and will take place in October this year. Thanks.
Phenomenal. Thank you, Valerie. The toolbox of being able to have a benchtop twin screw extruder is a game changer for researchers and for startups. Thanks for letting us know that ThermoFisher is in the space and willing to offer these products and services to people in the plant-based meat industry. Next up, we have our final flash talk. This is from Ciara McDonnell of CSIRO. She'll be presenting on protein under pressure. Protein under pressure. Welcome, Ciara.
Hi everyone, I'm Ciara McDonnell. It's a pleasure to be here at the Good Food Conference to present to you about the project Protein Under Pressure, High Pressure Processing for Improved Function and Flavour of Plant Proteins. And I'm delighted to make this presentation on behalf of the CSIRO project team. So just to set the scene on what is the CSIRO, well, it's the Commonwealth Scientific Industrial Research Organisation, known as Australia's National Science Agency. Quite a large organisation interacting with over 1,200 SMEs every year and globally renowned as a top research institute. And the food programme within the CSIRO is divided amongst two food pilot plants, one in Melbourne and one in Brisbane. And the core focus areas of the food programme can be seen on the screen here, one of which is ingredients and sustainable technologies. And within those, there is high pressure processing or high pressure thermal processing, which I'll speak more about today. So what is high pressure processing? Well, it's compression of a prepackaged food at 400 to 800 megapascals for less than 5 minutes and normally done at ambient temperature. And the main application of high pressure processing can be illustrated by this image here. So as you can see, at 100 megapascals, bacterial cells and small molecules like vitamins are unaffected. But as we move towards high pressures such as 600 megapascals, cell membrane permeabilization occurs amongst other things within bacterial cells. and the vitamin molecules are unaffected. So that leads to the main commercial application of HPP today. In fact, it's quite a mainstream application that we can inactivate pathogenic microorganisms while not affecting those small molecules responsible for nutrition and flavor, and really fitting with those fresh, natural-like products that consumers are desiring. But research has also shown another application. So another study out of the CSIRO has shown that in meat products like sausages, salt content can be significantly reduced by using high pressure processing to solubilize proteins. And there have been other publications from other institutes globally that show it can be used to reduce out the need for E-number additives in the label. So it depends on the parameters of the food material and the processing parameters of high pressure. But this is another interesting application because then it can feed into the clean label trend that consumers are also desiring. And interestingly as well, at the CSIRO, there are 35 litre and 3 litre pilot scale HPP units, but my colleagues have developed a patented 3-layer canister, which allows for adaptation of commercial HPP units. So then while the commercial ones currently operate at ambient temperature, this canister can allow for combinations with very high temperatures. And the main application is to inactivate spores, which ambient temperature high pressure processing cannot do. But with all of this in mind, that we know it can be used to solubilize proteins, and we know that new processing parameters are possible, we did wonder what is the impact on plant protein, and it is an area that is underexplored. And that led us to write for a grant to the Good Food Institute, which we received funding for this project, which we're very grateful for. So the project, Protein Under Pressure, really focuses in on using both high pressure processing at ambient temperature and high pressure thermal processing, applying that canister I referred to earlier, for improved function and flavour of plant-based proteins. And the overall aim really is that if we can affect function and flavour, there's less requirement for additives, and we can improve the quality of some meat analogues. So the project was divided into 4 steps. So firstly, selecting the proteins. We selected soy, lupin, faba and pea as our proteins of interest. And then we wanted to optimize the parameters. So as I said, there's many different parameters that affect different properties of the food material. So we looked at different pressure temperature combinations. And then we looked at the effect on technofunction. So we characterized the ingredients post-processing in terms of gel properties, texture, color, solubility, and water binding. And this project is just over halfway through, and the latter half is going to focus a bit more on the flavor. So today I'm going to present a bit of the findings around the results on effects on technofunction. So here you can see the results of HPP on protein solubility. So when we applied the high pressure processing at 200 or 600 megapascals ambient temperature for 2 minutes to soy protein isolate, we did have a significant effect on the solubility, especially at 600 megapascals. And when we used differential scanning calorimetry to see if this affected the denaturation, you can see here that the red and the blue line with the circle are similar, they're having similar enthalpy. So therefore, we couldn't really pinpoint what was the reason for this change in solubility. So then we applied low-field nuclear magnetic resonance to understand the water binding within the gel matrix. And as you can see on the graph here, the red line has a peak which is coming in shorter than the others, and that's corresponding to 600 megapascals. And on the bar chart, the grey bar shows that that relaxation time is significantly shorter. So what this indicates is that the hydrogen ions, those which are present in water, are more tightly bound within the gel matrix or more entrapped. So this indicates that we're getting improved water binding with the soy proteins after 600 megapascals of high pressure processing. And I did just want to touch on the other proteins that I mentioned, so lupin, faba, and P. On the graphs here, you can see we have our high pressure processing study at ambient temperature and our high pressure thermal processing study at 60 and 90 degrees Celsius. We have two temperature controls, T1 and T2, and high pressure thermal processing, one with 200 megapascals and two at 600 megapascals. And as you can see, really with that high pressure thermal processing study, we're getting a big increase in P protein solubility. So this really does warrant more research, but it's quite an exciting finding. So to conclude, there is potential to modulate plant protein functionality by application of high pressure processing, so these proteins can then be tailored towards food applications. And our studies indicate that at ambient temperature, 600 megapascals results in increased solubility and water binding. And preliminary data on other proteins indicates an interesting effect of high pressure processing on pea protein solubility. Our future studies within this project will look at the effects of the treatment on flavour. So the latter half of this project will be led by my former colleague, Dr. Regina Stockman at the CSIRO, and Regina can be contacted at this email address here. And I'm also happy to be contacted about the project. I'm still in close touch with Regina and the project team, but I have recently moved on to an exciting new alternative protein company here in Australia called Algae Foods. I'm happy to be contacted at the contact details on the slide. I'd like to say thank you to the project team for making this research a reality, and a special thank you to you for your attention and the Good Food Institute for the funding for this research. Thank you.
Phenomenal, phenomenal. Thank you, Ciara. With HPP, high pressure processing, being so widespread in the food industry, this is a phenomenal tool to add to the toolbox of modulating protein functionalities so that other tools besides enzymatic hydrolysis can be used to improve the solubility of proteins. Thank you for sharing that with us today. We now move into the second portion of today's session. This is on longer talks. We'll have two longer talks, 20 minutes each. First up, we have a longer talk from Shannon Hood-Niefer of the Saskatchewan Food Industry Development Center, presenting on innovating in food and plant-based proteins. Innovating in food and plant-based proteins. Welcome, Shannon.
Hello, everyone. I'm Dr. Shannon Hood-Niefer, Vice President of Innovation and Technology at the Saskatchewan Food Center. We're a not-for-profit organization that helps companies develop new food products and new food processes, new food ingredients, as well as some small-scale manufacturing. I'm here to talk to you today about innovating food using plant-based proteins. So this is an example of my favorite one, actually. This one is vegan beef, ginger beef, and it is delicious, and it's made from gluten and pea. And I'm going to talk to you a little bit about how we came to this in a generic way. But first, we'll start off with why the fuss? Why are we talking about meat analogs and plant-based proteins? This particular sector is growing at quite a quick rate, at over 10% compound annual growth rate until 2027. And why this high growth rate? Consumers are more aware that diet and nutrition affect health and wellness, and consumers are thinking more about environmental sustainability of where their food comes from. So what to make and how to make it? There's a wide range of ways that we can make different meat analogs. So using fermentation, so we can make tempeh or tofu. These are examples of that. We can culture meat, which is your lab grown meat, which is up and coming technology. And we have 3D printing, which is where you would print your vegan steak if you wish. There's also protein spinning and shear cell technology, which would use plant-based proteins and be able to fibrate them. But I'm here to talk to you about using extrusion to fibrate plant-based proteins. So these are the generic steps. So we have protein, starches, fibers, and maybe some micronutrients, some flavor compounds, and some color compounds in the raw material blend. And we're going to run it through the extruder. We're going to get what you see in the middle picture, which we consider an intermediate product. And so it comes out as a slab or a tube or whatever the die shape is. And then you need to further process that into a food product that would be recognizable by consumers. There are a number of challenges that need to be overcome in this particular sector. And so we have a picture here of some vegan chicken fingers for your viewing pleasure, but we have functionality. And so one of the things that we see is that different botanical sources of proteins obviously function in different ways, but then there's also the compound added challenge of that, how you've extracted the protein and maybe the thermal history of that protein. So the different time temperature combinations through drying potentially will have an impact on that functionality. And those functionalities may or may not lend itself to fibrating using extrusion. And we do find that extrusion, especially high moisture extrusion, is a bit more sensitive to these subtle, the subtle thermal history of different proteins. And so, you know, what might work well in where we're seeing, I guess, a narrower tolerance of functionality in high moisture meat analogs. And so that's something to consider here too. Appearance, you know, obviously we're trying to make this look like meat in this particular case. And obviously there's applications of meat alternatives where we don't want it to look like meat, but that's not what we're talking about today. So we want things to look like meat here. And then flavor, you know, plants taste like plants. And we're trying to make something that could potentially be bland so that we can add in different flavor compounds. And then, you know, how do we detract from some of these plant-based proteins? Some of them are bitter, some of them are beany in background notes. And so we might need maskers. And then, you know, the great thing is that we're working with different flavor houses that are developing these really wonderful flavor compounds and these maskers to help this particular case. This is vegan calamari, and it's also quite good. This might be my second favorite of the vegan products that we've developed. Color is also a challenge. And so here we have, you know, meat comes in a wide range of colors. We've got different shades of brown and red and white and gray, not so much gray, but sometimes depending on how you cook it. So and then meat color changes through the cooking process. And so how do we mimic that as best we can? But the biggest one is texture. So how food feels in your mouth. And so we have, you know, and so we've been here at the Food Center, we've been doing high moisture meat analogs since 2008. And, you know, and we've made some really fantastic things, but we've also failed miserably at times. And so we've made really gritty things, you know, meat alternatives that wouldn't be considered a meat alternative. They're too chewy, too gummy. You know, and then how do we measure and look at degree of fibration and the comparisons between trying to mimic different meat products, the direction of fibration, which is important when you're trying to do steak. But the real big thing is that juiciness, that succulency that you get from a meat product is really challenging to mimic using extrusion because of the lower fat content that we're working with. And so how do we introduce all these different compounds to deliver the texture that consumers are looking for? Cost can be a bit of a challenge here, depending on the ingredients that are being used. Also, because this is an emerging field, and in a lot of cases we're using emerging ingredients, we're looking at, you know, some ingredient shortage, and especially now during these times, we are seeing ingredient shortages and transportation issues, to be entirely honest. And so we are looking at alleviating some of those. And then because this is also an emerging space, there isn't a lot of places for companies to test their processes in a food safe place where they can actually deliver product to a consumer for consumer testing panels. And so we help with that whole development phase here. We also see consistency in some of the ingredients, especially the up and coming ingredients where there's a lot to lot variation, whether it be some of that thermal history or maybe some particle size variation. Even though the protein content might be exactly the same lot to lot, we are seeing some slight differences in how it functions during extrusion. But invention and innovation has to start somewhere, and we've been doing this for a long time. And so I've been here at the Food Center for a dozen years, and we've been doing that and then some. So I'll begin with ingredient breakthroughs. And so over the last little number of years, we've seen synthetic heme come out. We've seen some great introductions of new functionalities of ingredients, some new fat sources, and some really great flavor compounds that are really helping this particular thing. And so if I was to set a high bar, this photo is a real meat photo. And so this might be something that we would be looking to attain. So some marbling of fat in meat, and how do we get that color change to mimic a rare or medium rare steak. So I'm here to talk to you about extrusion, and there's two types of extrusion that kind of interplay here. So we have dry texture plant proteins, which are shelf stable post extrusion, and it needs to be rehydrated to use. And then we have high moisture meat analogs, which is the bulk of what I'm about to chat about here. So ideally, we're looking to take a plant-based protein, which tends to be a bit more globular in nature, and then we're going to denature it and unravel it, and that hopefully it aligns with the flow of the extruder, and then as it cools, it binds to the other protein molecule that's also aligned, and then it binds to form these strands of proteins. And the degree of which that binding happens plays into the degree of fibration. So this is the bulk of what I'm going to talk to you about today, and this is a really full slide, so I'm going to break it down just a little bit for you here. And so we have a number of levers that we can pull to control finished product quality using extrusion, but the biggest one is raw material selection. So the type of starch and the content of starch, the type of protein and the content of protein, fat, water content, and particle size all play a role in the finished product quality parameters. I'm going to pick on protein for a bit. As an example, if you were to add even the tiniest bit of wheat gluten, you would expand the range of functionalities and the textures that you can achieve with an alternative protein. So if we were to say blend pea protein with a wheat protein, even that tiny bit of wheat protein, say maybe 10, 15%, 5% of a raw material or a dry mix going into the extruder, you could create a much wider range of textures. Lipid content also plays a role, and so there's a wide range of ways that you can get lipid or fat sources into the extruder. So you can pump it, you can add dry, dehydrated fat sources, or you can try adding it later. We find pumping to be a pretty good way, but we can't get up. We're working on ways to get it up higher than what we currently can. How that extruder is set up also plays a significant role in the finished product quality parameters, and things that we would adjust depending on texture, formulation requirements, screw profile, how aggressive or how assertive that screw profile is, where your mixing elements are. Do you need to pump in something like a fat source later on in the extruder, so you need to create a bit of a low pressure zone? These are all things that we would consider when we're developing a screw profile. Die design, and right now in high moisture meat analogs, a lot of the dies are rectangles, and so you're making a rectangular shape that needs to be cut. So how can we maybe improve on that or innovate new dies? Then the cooling rate through the die, this also plays a role in terms of how strong that fiber is set. And then some companies use preconditioning in high moisture meat analog extrusion and some do not, and so that also plays a role and might be predetermined by the protein source that you're choosing. Extrusion operating conditions, this one is probably the most fun to play with because it's the biggest one that you can have impact on the day that you're running. And so screw speed, throughput, thermal energy, and when and where you put moisture does play a role. And so sometimes when you're extruding with a preconditioner, how much moisture you add in the preconditioner, the temperature of that moisture, and then how much moisture you add in your extruder can adjust your finished product quality parameters. And all of these things impact screw fill, melt temperature, pressure in the extruder, your residence time, I've got residence time up here twice, shear or your specific mechanical energy, viscosity in the extruder, which also impacts pressure, and the torque. And so how much energy that extruder needs to put in to turn those screws. And then all of these things impact final product quality, texture, expansion, any texturization, water holding capacities, nutritional profiles and nutritional properties. And so we do see that sometimes we may or may not lose some lysine through this process. And so we might need to augment with maybe a bit more pulse to make sure that we have enough lysine to have a complementary protein. We also might be looking at adding or fortifying with different vitamins and minerals to make sure that we're meeting any nutritional qualities that we might be looking for, the consumer might be looking for. So we might need to control and test after to make sure that we're hitting the targets. Finished product quality shelf life, how long that product lasts. Is it refrigerated or frozen, and how long that lasts, and what type of package it should go into. And stability of color, shape, and flavor. So impact of food processing. Obviously, we want this to be shelf stable as much as we can, but food safe. And so we treat high moisture meat analog like real meat. And so we ran it through a full food safety program like we would a meat process. Extrusion in this case does impact starch gelatinization and protein denaturation. And in this case, plants, we're trying to impact the digestibility and the bioavailability of those proteins. So there might be some really interesting ways on the ingredient innovation side. Maybe fermentation of some proteins prior to extrusion might help extrusion, but it might also help with the bioavailability of those proteins post-extrusion. Obviously, hopefully they're more palatable after, and that we've developed different flavors. And so we're looking at, different mechanisms to add flavor in and retain that in, and both in extrusion and post-extrusion, and then texture. And the reason why I do this is for diversity of food products, so that there's more choice. So when you go to the grocery store or to your restaurant, that you have a great, well, not too much choice, but a great deal of choice, so that you can get the flavor and the texture and the eating experience that you're looking for. And as I said, the extra date is an intermediate product. And so sometimes we do some size reduction. And so you can break it all the way down. So you can put it in a chicken nugget, which would be the smallest size here, or you can cut it into different chicken pieces. I call them chicken tenders or beef tenders, so that you can, throw it in a stir fry all the way to, you can cut a chicken breast or a steak out. And then, so from our product development team here, which they've done a ton of work on alternative beef patties. And so we have here a picture of some real meat patty, and then a traditional veggie patty, which wouldn't necessarily be mimicking meat so much, but has more vegetables and, say, maybe rice or quinoa, and some, you can see some corn and carrots in this particular picture. And then we have the meat patty that's looking to mimic a real meat patty. And this has also come a long way. And so this is a picture from our lab of our alternative meat patty. And we've had a great deal of fun, I guess, in developing new food products. So we've got some pulled pork here, the burger, some chicken stir fry, and a roast beef here, in addition to some breakfast sausages, some pork, some chicken nuggets, tuna, and then a battered shrimp-like piece, and then some pork ribs. Obviously, we've been putting a considerable amount of time into pepperoni sticks and deli meats and sausages, which is incredibly challenging from a processing perspective, because you're also trying to, so you have your meat bits, but then you're also trying to make a gel and a plant-based emulsion that is not dry or mealy, which we've come up a long way since then. So at the Food Center, we have access to over 300 pieces of food processing equipment, and we have 50 people, just over 50 people here that have been technically trained, and we look forward to chatting with you. And my apologies, I wasn't able to be there in person today, but I'm happy to entertain questions via email, should you wish. Thank you, and have a great day.
Thank you, Shannon. It's phenomenal to know that Saskatchewan is playing such a huge role in plant-based meats, and that the Saskatchewan Food Industry Development Center is available for startups and entrepreneurs looking for product development advice. Next up, we have our second longer talk. Our second longer talk is from Theresa Rothenbücher of Revo Foods, presenting on the next generation of fish plant-based addition. The next generation of fish plant-based addition. Welcome, Theresa.
For the kind introduction, Ryan. So as Ryan said, I'm going to present how Revo Foods is using 3D printing technology to produce plant-based seafood. And first of all, I would like to give you a little introduction about Revo Foods. Next slide, please. So Revo Food was just founded this year in January in Vienna, Austria. As of now, we have 13 employees, and we have two different main lines of R&D. First of all, the food technology side, where we develop the recipes for different plant-based fish products. And secondly, working closely together with our hardware development team. So this is a real specialty of Revo Foods that sets it apart from other startups or companies in this field. We develop our own 3D printing technology and use this to innovate new products that are not yet available on the market. Next slide, please. So who's hungry? Let's take a look at these nice fish pictures here. On the left, we see smoked salmon or raw salmon. And as we see, the appearance of raw smoked salmon is very shiny and almost gel-like. And we can see that there's a structure. It's not a homogeneous product. It has these white stripes, which is the connective tissue where the fat is located. And also, when you tear it apart, it breaks at these white stripe parts. And also what is very special about this raw fish, that it's almost transparent. When we look at the cooked version of salmon, however, it is more opaque, and it's also not anymore a gel-like texture, but more flaky. So we are not talking about fish sticks here and not talking about fish burger, but really these fully structured fish products. So next slide, please. How are we actually reaching such a plant-based product? So first of all, of course, we need to choose the right ingredients that resemble the nutritional value of fish, because this is why consumers love fish so much. Not only the taste, but also that it is rich in proteins and has high omega-3 fatty acid levels. So of course, we have to choose the right plant-based protein sources and combine them with omega-3 rich oils. It can be plant oil, flaxseed, or microalgae oils, depending on the region, what is allowed to use. But then also the ingredients should not only be nutritious, but they have to kind of resemble the taste and appearance of fish. So it would be great to add some algae if you want it more natural, or of course you can also add some flavors that make it closer to the real thing. And for the appearance, it will be very important to choose materials that are kind of, if you go for raw fish, kind of transparent, for example, when they are dissolved in water. And you have to make sure you need to combine them the right way to actually reach a texture that comes close to the product you desire. And if you have the right ingredients, of course, it's also important to choose the right technology that has the precision, because we have seen some of the extrusion presentations before, and it works super, super nicely to texturize products for meat like chicken. But fish has a really different texture, and it's not as homogeneous. So we need technology, production technology that really precisely can mimic these white stripes within the product. But we don't want to make it too perfect. It should still look natural and tasty. So we don't want to have a rectangular block with sharp edges. So it would be great to also have different designs, different versions that are looking good, but not too perfect, not unnatural. So in the center of all these needs is the technology. that can create or can put all these needs together. And this is what Revo Foods is working on. Next slide, please. Can you please start the video? Perfect. Yeah, so without the volume, please. Thanks. Perfect. So yeah, now I'm going to show you a little bit of the concept, how Revo Food is going to produce their products. So this is not actually how our printers look like, but it is a conceptual video. So of course we start off with some nutritious ingredients, as I said, some proteins, some algae for the texture, and plant oils for the omega-3. And those ingredients are mixed together. Here the example of salmon. So we have some orange material, some white material that is continuously led to the printer and also in a continuous way fed to the 3D printer, to the extruder. And here also continuous process is leading to the nozzle, and then we have the 3D printer printing the product in a very coordinated way. And then it's ready for packaging, basically. Next slide, please. So this is what one of our first products look like, actually. It is plant-based smoked salmon slices. I hope that you have good quality so that you can actually see the 3D printer lines that resemble also very nicely the lines of or similar to salmon. We are aiming for high quality salmon products that are kind of mimicking the wild caught salmon type. So not a lot of fat tissue. So we actually do not really need to include white parts here to mimic the appearance. And I can assure you that the taste is really, really good, because we had some vegans that even said it's too close to the real thing. But what about the texture? Next slide, please. So we did some texture analysis to be super sure that we are close to the real thing. And on the left, you see some parameters that we chose. It was a TPA measurement. And we had 3 different samples. One was a conventional salmon fillet. Then we had here in light gray and dark gray, you see conventional smoked salmon slices. And in the middle in white, you see our 3D printed plant-based salmon slices. And all samples had the same dimensions of 2 × 2 × 0.5 centimeters, so that we can compare it nicely. And yeah, as you see for the hardness, our plant-based 3D printed product is somewhere in the middle, so it's totally in the range of salmon, somewhere between the fillet and the slices. Next slide, please. And also when we check the adhesiveness and elasticity, we see we are very close to the conventional smoked salmon, even closer to the smoked salmon than to the fillet, to the raw fillet. So that's pretty good, because, for example, with the adhesiveness, if you have a smoked salmon, a conventional one, in your between your fingers, and you kind of touch, and it feels kind of sticky. So we were able to mimic this stickiness as well for our product. Next slide, please. And also for the resilience and the cohesiveness, we are kind of in the range of the salmon. So we were really happy to have this also confirmed with data, not only with our sensoric or mouthfeel or people we ask about their opinion. So also the science behind it says we are on the right track here. Next slide, please. So now the question, how do we actually get from the prototype printing to a real production facility for plant-based fish products? Could you please start the video? I think it is without music in this case.
There's no video on this.
If the video is not working, I can just explain.
Yeah, there's no
Okay.
video, it's not working.
Okay, A shame. It's a cool video. It's a cool animation. So, but you see on the picture anyway. So the plan is, or we are currently working on connecting several printers with a conveyor belt. So that all these printers are working in parallel and are very well coordinated. So as soon as a printer is finished, it pushes out the tray, and the tray is transported to the packing station. And one of the main advantages of this is also if one of the printers is for some reason not working, we can just disconnect it, repair it, maybe even replace it with another one, and the whole production line is still continuing while this is happening. So, for example, if you have these huge extruders, which has a really high throughput, but on the other hand, if something is broken, the whole production has to stop. So also we can stack these production lines on top, so it can be really saving also a lot of space. And yeah, next slide, please. So if you're interested in what we are doing at Revo, we are always happy to collaborate. So please don't hesitate to contact me at Theresa@revofoods.com. Or if you're even more interested, I'm happy to tell you that we are hiring. For example, we are looking for a senior food technologist. So you can directly hire me at revofoods.com or check out our webpage, revo-food.com/join us. And I'm really happy to take your questions now if there are any. Thank you very much.
Phenomenal. Thank you, Theresa. I can't wait to try this product.
Yeah, actually, if you come to Austria, you can already try it. It's already in the restaurant here, and soon will reach the retail, the supermarkets.
Phenomenal. We'll now take a 5-minute time slot. And since Theresa ended her presentation early, we're going to have a 5-minute time slot for audience questions. Please enter your questions for Theresa in the session chat. Also, attendees can hit the thumbs up, like button to upvote a question, and we'll get to as many as we can. Theresa, I'll start off with some of the first questions in the chat here from James. Theresa, traditional 3D printers are a notoriously expensive way for producing plastic products. How are you going to scale this approach for salmon?
Okay, well, as I showed on the slide, we are going to really not work with single printers, but building up a printer facility. So this is something that, to my knowledge, does not really exist yet for the food industry. So we are really developing this upscaled food production line. And we still have to prove that it works, of course, but we are in a good way right now. I hope that answers the question. I'm also sorry if I cannot go too deep into some details because it is also some intellectual property. We have some patents pending, and I try to answer as well as I can. I hope that's fine.
Thank you, Theresa. Next up, from David. Is this tech suitable for large-scale production or for at-home customized production? For example, how long does it take to print and cost?
So the food printers, so we also try some of the food printers that are available on the market, and they are quite slow. So what we are doing, we really want to speed up the process. We develop our own technology for this. And I think the market, maybe in some countries, I don't know about America, for example, maybe people would be ready to have their own printers at home, but this is not our main target. We really would like to produce large-scale salmon products and then sell them to the retail markets, and then people can buy them there. That's single printers at home is more like a gimmick or something for the future. It's not right now.
Thank you, Theresa. Next up, everyone thinks this is a great product. And the question is, how do you eliminate the green color from algae? Do you use food colors in the yellow, orange, and white portions of the salmon?
Yes, so as I said, of course, you have to choose the right ingredients for that. And then it makes sense to choose ingredients that are neutral in appearance so that you don't have too much off color from when you start. But then you have to choose colors, natural colors. There are a lot of natural colors available and even very, very good ones. So the market for this is huge. And then you have to find the right color and also make sure that it's kind of stable for pressure, heat, and all these kind of things that might come up in the process. And yeah, we definitely use colors to reach the right product appearance.
Thank you, Theresa. We have one last question. We have several attendees that have asked, what is the shelf life? What is the shelf life of this printed product?
So I'm very happy to say it's much better than for fish. So I think fish is like 2 or 3 days and then you shouldn't eat it anymore because it can be quite dangerous. For us, we can surely say 3 weeks in the fridge is fine. We know that you also can freeze it for a very long time and thaw it without quality loss. And we want to reach a shelf life of 6 weeks eventually.
Phenomenal. Thank you, Theresa. Audience members, that concludes our Q&A session for today. Theresa, thank you for joining us, and I can't wait to try Revo Foods plant-based salmon.
Thank you very much. It was a pleasure. Bye-bye.
The final part of today's session is a panel with Mario Martinez of Aarhus University, Girish Ganjyal of Washington State University, and Birgit Dekkers of Revo Foods. Welcome, Mario, Girish, and Birgit. Mario, Birgit, thank you so much for joining us today. As we're waiting for Girish to join us, could both of you please introduce yourselves? What's your name and what is it that you do? Birgit, we'll start with you.
Sure, thanks for the invitation. My name is Birgit Dekkers and I'm one of the founders at Revo Foods. Revo Foods, we develop a new technology to structure plant-based proteins, which is called shear cell technology. And that enables us to create larger pieces of structured material for the application of whole cuts.
Thank you, Birgit. Mario, over to you.
And this is Mario Martinez, and I am an academic survivor, I would say. I am assistant professor at the Aarhus University at the Department of Food Science, and I have been working on this, on extrusion for many, many years, usually low moisture extrusion. But then I found the opportunity to transfer a little bit of the extrusion knowledge into this exciting area of developing HMMAs, and we are working on that.
Thank you, Mario. As a note for our attendees, there will be a brief question and answer session following the panel. I encourage everyone to submit questions in the session chat window throughout the presentation, and we'll work through those at the final Q&A session following the panel. Next up, since we've got such an array of perspectives here, let's dive in to our questions for today. First, Mario, I'm going to start with you. Mario, your research has focused on texturizing plant-based meats at the micro scale. How can we advance the texture and structure of plant-based meat using extrusion technologies?
Well, I would say that's a difficult question that has, and I would say it has many answers. One of the main things that I would say, but I am biased obviously, is we can advance it by using extrusion. I know that it is, the word extrusion was also in the question. Why I'm saying this? Because we have to understand how different proteins will behave under under specific processing events. When we're using extrusion technology, for example, it's not just a matter of heating and cooling, It is also a matter of subjecting the unfolded proteins to the right flows, for example, elongational flows and tensile stresses. So this is sometimes necessary in order to promote anisotropy. So understanding this, I think extrusion could have an advantage over other technologies. Now, I am completely biased because this is the only technology I have been working on. I would say that we can, one of the main opportunities I would say that we have is, well, first of all, understanding the materials that we are working with. And I am more fond of using concentrates and dirty stuff rather than isolates, because we have to consider everything. We have to consider costs. We have to consider as well environmental fingerprints. And I think the future is more brilliant for working with dirty stuff like protein concentrates. But in order to do that, there is some challenges associated with that. So we need to understand many things of them. We have to understand how the proteins will behave, how the proteins will compatibilize with the rest of the stuff, mostly carbohydrates. We have to understand as well that we're working with plants. At least in my research group, we are just working with plant materials, plant protein concentrates. And there is a problem. That is that there are many plant primary and secondary metabolites that will significantly affect the protein functionality. So understanding everything and getting a little bit of understanding of all this mess from a compositional perspective, but also from a structure function perspective, I think it will provide plenty of opportunities for the development of good textures. It's not easy, but we will see how far we can go.
Phenomenal. Thank you, Mario. And that's such an important question to get right, is that concentrates over isolates can significantly decrease costs for manufacturers. And knowing what happens to those carbohydrates in those concentrates will help produce even better products for consumers. Birgit, my next question is for you. Your company is tackling plant-based meat extrusion in new and interesting ways. What do you see as the biggest challenge in plant-based meat extrusion right now?
Yeah, I think, thank you for that question. I think one of the most important things is getting consumers into these products. Of course, the market is growing very rapidly, but it's still quite small. So we have to address more consumers. And the way how you can do this is by improving the quality of products. So there are already many great products out there today with great texture, taste, and bite. But I think we have to expand the product portfolio that we have to address the wishes of more consumers. And what we try to do with our technology is indeed a variant or a different way of extruding or transforming, structuring proteins as you wish, is creating different types of textures, creating more juiciness, and expanding the product portfolio that we have at the moment in the plant-based space. And I think that's one of the most important challenges that we have, is expanding the products that we can create, expanding also the proteins that we can use. I fully agree with Mario on the use of protein concentrates, isolates, maybe in the future even flours. That will be very important indeed to decrease costs, but also improve the sustainability of these types of products. And understanding better how these proteins interact in structuring processes to create in the end better end products. That is very, very important.
Thank you, Birgit. I can't agree more. We've got to expand the portfolio of textures that are available to consumers. And it's these new processing technologies that are helping develop that portfolio. My next question, back to Mario. Mario, what are the most critical changes in plant protein extrusion we must make to face the future of all protein effectively? What are the most critical changes in plant protein extrusion that we must make?
Well, I would say that the future will well, hopefully it is not like that, but I believe it will be like that. For example, climate change will narrow the array of possibilities we have, or the array of protein sources we can work with. So then even once we get, for example, a great understanding of a specific protein that will work perfectly, then perhaps that plant will not grow anymore at the quantities that will be needed to produce at large scale. So then we will have to reinvent again, and then to try to work with new protein sources that are less unknown and all of that. So I think we will have to get used to working with proteins from climate resilient crops. Excuse me, and that will increase a little bit the challenge, I would say. I don't know if I answered your question.
Thank you, Mario. That's an excellent point, is that climate change will narrow the number of plants and ingredients that are available for processing. So it is critical for us to be able to develop new technologies, new processes to make sure that we can get the right type of protein to the consumer. Next, Birgit, who is making the greatest advancements in plant-based meat extrusion, and what are they doing? Who's making the greatest advancements in this space right now?
Of course, I have to say we are, but no, I think there are many companies and universities that are working in this field and making great advancements. Of course, early on in the value chain, there's indeed the protein ingredients that are being created. Today, most of the products are created with soy, pea, and wheat proteins, but the number of proteins that can be used in structuring technologies are expanding very rapidly. And I understand your concerns, Mario, with the number of proteins that are available to us due to climate change. That is definitely a thing. But I also think that there are a lot of companies and researchers investigating how we actually can expand the protein portfolio that we have and structuring that into great end products. So I think there is also, I'm very positive that actually the number of ingredients and proteins that we will use in the future will actually expand from plant sources, but also maybe from other sources. So think about microproteins or other sources of protein that can be structured with different technologies, like extrusion, but also other technologies like 3D printing, and what we use is shear cell technology. I think it's an entire field that is moving and making advancements, and therefore I don't think it's just one party or one company that is making a big change. I think we are all doing it together, and I think this event is also a great way to collaborate with all the parties that are active in this field to advance this space.
Thank you, Birgit. And I think that's perfectly acceptable to respond that you feel it's your company that's making the greatest advancements in this space. Next, we'll go back to Mario. Mario, your research has focused on extrusion of interesting combinations of plant-based proteins and their carbohydrate fractions. What do you see as the most interesting trend? What's the most interesting trend in plant-based meat extrusion for 2021, this year?
Well, I don't know if a trend or something that everybody is looking for is to try to simulate a whole muscle cut, a beef-like whole muscle cut. That could be, I think that is quite challenging, but that is something that all of us are really looking for. So it won't be easy. This is one of the things that we are trying to do in our GFI funded project, and we are getting some results. Some things, but obviously still is a little bit different, and I do not know if we will be able to perfectly emulate a whole muscle animal tissue. But the question is, do we really need to emulate that? Can we develop perhaps a new material that consumers will even like more? That's a glooming question there that we should wonder.
Thank you, Mario. That is a critical question for the industry, is how do we move towards whole cuts? And just like the announcement today that GFI is giving an additional $5 million in funds to the research grant program to help support top scientists around the world in their quest to achieve whole cuts. Birgit, for the next question, we're going to come back to you, specifically your company and how you got involved there. What made you decide to tackle plant-based meat structuring? How did you get into the industry, and why is it that you stay?
That's a good question, and I think it is really interesting to work on our changing food system. So I really believe that there is a transition from animal-based towards more plant-based food, and there's an urgent need for many people to work in this to make it happen. So I have a background at Wageningen University in research, where I did a Ph.D. on structuring proteins with new technologies, led by Professor Adriaan van der Groot, which was very, very interesting. But I also saw that there was an opportunity, or basically there was a bridge to gap between science and actual commercialization and development of products, and commercializing them to make it available to consumers in the end. And that's what we're doing today with Rival Foods. And I think that is very exciting to really go from fundamental research to commercial implementation and development. So I would also like to advise all the people that are in here that are in science and wish to have more impact, that is also a very interesting journey to develop the science that is underneath very challenging problems that we have today in our food system, to take that journey and take that step and start implementing and developing the science.
Thank you, Birgit. That is extremely true. There's a huge need for talent in the sector. And when thinking about different fields of research that young STEM professionals can go into, alt protein is a leading example in terms of counterfactual impact. Next, my question's for the entire group. What's the question you are most tired of hearing on extrusion? What's the question you're most tired of hearing on extrusion? And what would you like to say about it so that you never have to answer it again? Mario, we'll start with you. Mario,
Yeah,
you're...
Yes, can you hear me now? I don't know if I have one that I would pick, one favorite one. I guess I have heard many. One of them is well about extrusion. If these materials that we develop with this laboratory scale extruder can be scaled up to large scale production. This is one of the questions that I hear the most. And I will answer that, and I can answer that with experience, because we have one of a very small extruder, and also we have a large extruder. And I would say that both are quite interesting to work with, because of course what we produce with a small scale extruder is not scalable. So this is the answer is clear. It's not scalable. But the amount of understanding that we can get using that small one is also unique, and we cannot get it with the pilot scale one. So the fact that a small extruder is not scalable, that doesn't mean that it's useless. No, it's also quite interesting to have it. So this is I would say that this is the answer I have with that. And this is also this also brings up the point that in this world, both chemists and engineers, we have to be working together, because we really need to understand what is going on for example, in the protein unfolding, protein in the interactions after extrusion, or the effect of tensile stresses at, for example, elongating a dispersed phase that then results in a fibrous structure. So but also we chemists also we need to understand, for example, how to I don't know, how to design a piece of equipment, such as, for example, rival foods, such as a piece of equipment that can then be working and resulting in interesting materials with interesting properties. So we have to work together.
Thank you, Mario. I will make sure that I never ask you again, is it scalable? Birgit, over to you on this question. Which question are you most tired of hearing on this subject, and what would you like to say about it so you never have to answer it again?
Yeah, I was happy that you gave me the question already before this actual panel started, and I've been thinking about it a lot, but I haven't came up with the answer. Although I really like the answer that Mario gave, because we often get that question as well: is it scalable? And how are you going to feed the world with plant-based proteins at scale? And it is something that we are challenged with, of course. I think the entire field is challenged with how are we going to scale further so that, well, the balance between animal-based protein and plant-based protein is back to what should be for climate, for sustainability, but also for health. So I think indeed it is a question that is tiring, but at the same time, it is also the most important question I think there is to really make a step forward in this space.
Phenomenal. Thank you, Birgit. And I couldn't agree more. The levels of production for different foods that we have with conventional animal agriculture is not sustainable. And we do need to go back to different ways of producing food in a way that is a sustainable system for the earth as a whole. We have time for one last question before we transition to our question and answer session. And this is about, have we left anything out here? What's the elephant in the room here? Birgit, we'll start with you. Is there anything we're leaving out here that needs to be addressed?
It is a very good question, and actually it was already mentioned by Mario, I think, is the fact that what are we developing with extrusion or with other processes and technologies, and do we need to mimic the animal-based properties, the sensorial properties completely, or are consumers changing their habits by consuming more plant-based proteins and plant-based protein foods? And how is this going to evolve? I think that is one of the most important questions that we will be facing as an industry as a whole. I think a lot of companies, even ourselves, are focusing on flexitarian consumers, so addressing really sensorial properties which make a product similar to the animal-derived one. But how does the consumer look at these types of products 10 years from now? I think in food technologists, we are quite creative, so we can create a wide variety of structures. We can create a wide variety of flavors, even of colors. And how is the consumer looking towards these products in a couple of years from now? Would it be okay if we use green proteins, which have great nutritional value, but make the product having a great green appearance? I think those questions are on our Friday afternoon experiments often raised. How are consumers looking towards these products in a couple of years from now?
Thank you, Birgit. And that's an excellent point. We do need to take the long view on consumer acceptance of these foods, with sensory properties being a primary driver of acceptance. Mario, over to you for this one. Is there anything we're leaving out here that needs to be addressed?
Well, I really like the answer from Birgit. It's a quite interesting response. And also I see in the chat that Chris Menzel mentioned that we have now Pringles that are not potato chips, are another thing, but people love it. So it's similar to this. It's a new product so we don't necessarily need to completely emulate what a whole muscle tissue is, but perhaps we can develop new things. And I would like to add just another elephant in the room that we didn't mention, is that is nutrition. Is the anti-nutritional factors that are usually in many of the plant proteins, and also the nutritional quality of the proteins. So it will be important to work also with blends that have a balanced amino acid profile and that also have a good protein bioaccessibility. So that will be also quite interesting. And working with, I said before, dirty stuff, working with protein concentrates, even protein flours, but we should pay attention a little bit to the anti-nutritional factors. Some of them can be degraded during extrusion, for example, but not completely. So there will be, in that scenario also, I think we will also need to resort to plant breeders to try to get new breeds, perhaps with lower content of anti-nutritional factors. I will just add this as an elephant in the room.
Thank you, Mario. That's an important point to bring up from the chemistry standpoint. What are the anti-nutritional factors here, and how is that going to impact consumer acceptance and the nutrition of the end product? Team, we're now going to move into the final Q&A portion of today's session. I'll be going through the questions that have been submitted in the session chat. As a reminder to our audience members, you can submit questions in the session chat and upvote the questions of others in the session chat. Our first question from the audience is protein chemistry. Can proteins be made through chemistry rather than from plants? Can proteins be made through chemistry rather than from plants? Who would like to field this one first?
I mean, I can say that yes, they can. If you ask me how, I have no clue, because that's not my area of expertise, but I know that they can be synthesized. The problem there, I think, it will be the cost, probably as compared to other proteins from other sources, such as, I mean, for example, algae or unicellular, such as chlorella, unicellular microorganisms, or plants. Perhaps they will have a prohibitive cost. But the point is, this is just my point of view. Okay, we can make them, but I mean, we don't need to make them. Let's a living organism to make it for us, and then we just take it or extract from any source, such as, for example, from chlorella.
Thank you, Mario. Next up, we have a question from Varun. Commercialization of new technologies beyond extrusion is a major challenge for companies. What are some of the imperatives to be defined in the process of developing whole muscle cuts? What are some of the imperatives to be defined in the process of developing whole muscle cuts? Birgit, over to you for this one.
Sure, thanks. So one of the most essential things in whole cuts is the structure of a muscle, of course. And that results in a certain mouthfeel, a certain bite of products. And that's what we are doing, realizing with shear cell technology. We're structuring plant proteins in such a way that they give the same bite and mouthfeel. One of the things that what a muscle has is anisotropy. So that means that the structure feels different in different ways if you cut them, but also if you chew on them, if you bite them. And that's something that we are focusing on. Of course, this is one of the things that a product has in terms of a whole cut. There are many other aspects that are very important as well, such as flavor, color, the amount of juice that it can release when you chew upon them. But I would say that texture is one of the most important ones.
Thank you, Birgit. Agreed. Texture is one of the most important things for plant-based meat. Next up, we'll take a question from Michael. And this is for Birgit. Is continuous processing possible for shear cell, or is it a batch process? Do you consider this a challenge for scale up? Birgit, over to you for this one.
Yeah, scale up is definitely necessary. So if we want to feed the world with more plant-based foods, scaling is definitely an important challenge that we are facing with our technology. How you do this, it can be done in different ways. You can create a semi-continuous output, and you can make it continuous. There are different approaches to it. But in the end, the most important thing is to scale up so that we can have throughput sufficiently enough to feed more consumers. And that's what we're working on at Revo Foods.
Thank you, Birgit. We'll take one last question from the audience. This question's from Lynn, and we're going to direct this to Mario. Mario, based on your research, what type or mix of plant-based proteins make up the best amino acid profile similar to meats? Mario, your thoughts on this?
Well, we focus mostly on texture and the nutritional, everything regarding nutrition in terms of carbohydrate and polyphenolic compounds, but we haven't studied protein digestibility. But what I can say in terms of just amino acid composition, I can say that combining, for example, some proteins from cereals with some of the proteins from oilseeds, then we can have a very nice balanced amino acid profile. Now, in terms of protein bioaccessibility and digestion, then I don't have much insights about that. There are some reports that say that, for example, thermomechanical processing improve, for example, extrusion processing may increase protein bioaccessibility or digestibility, but then we have to pay a little bit of attention at the different interactions after processing, because that can make the proteins not bioaccessible, and then it doesn't really matter the amino acid profile. The proteins won't be digested. I think there is still a lot of room of research in this area, and we must pay attention to this alongside the texture.
Thank you, Mario. All this concludes our panel session for today. Thanks to Birgit Dekkers of Revo Foods and Mario Martinez of Aarhus University for joining us today. Finally, thanks to all of our panelists, all of our speakers, and all of our audience members for participating in today's session, Product, Equipment, Innovation. Thank you again. Signing off.